Proteostasis governs differential temperature sensitivity across embryonic cell types by Kaylee Vasquez, Alejandro Mercado, Victoria Flores, and Mary Both. Introduction. In this paper, we seek to understand whether cell types respond differently to temperature stress and what mechanisms contribute to differential response. In this research paper, proteostasis governs differential temperature sensitivity across embryonic cell types. We investigate how temperature affects different types of cells during embryonic development in the zebrafish. Temperature plays an important role in embryonic development across animal development. With an increase in temperature, studies have shown that development may accelerate by increasing metabolic rate and protein synthesis. However, temperature increase can also lead to protein misfolding and aggregation. There are many key terms to note in this paper, one of those key terms being proteostasis. Proteostasis is the maintenance of proper protein folding, synthesis, and degradation. Proteostasis is critical in embryonic development and may be disrupted with environmental stressors. Framework for the problem. By studying the response of different cell types in zebrafish embryos, research may determine how cell types maintain proteostasis and defend against temperature stress. The researchers found that different cell types within the developing embryo had varying sensitivities to temperature change, which could lead to developmental abnormalities if not properly regulated. The significance of this study is that this study may contribute to find strategies for the improvement of embryonic health when faced with environmental stress. It may also help with identifying mechanisms of development robustness. Another key term in this paper that is important to take note of is robustness, which is the ability of a cell or organism to keep normal development despite environmental stressors. This study may help in the field of toxicology or in the field of developmental biology. An example of the importance to toxicology is that it may help us understand the mechanisms underlying sensitivity to environmental stressors that can pose risk to embryonic development. By understanding these mechanisms, we can come up with a strategy to mitigate those risks. It may be also helpful in the field of developmental biology as we learn how cell types within an embryo maintain proteostasis and may help us maintain healthy cells. Why are zebrafish embryos used in this study? Zebrafish are used because they naturally experience temperature fluctuations in their environment, which makes them a good model system for studying the effects of temperature stress on embryonic development. Zebrafish embryos are naturally exposed to temperature fluctuation. They display a temperature-induced phenotype and alteration in development rate. However, the mechanisms of how cell types buffer against the effects of temperature stress are not fully understood.
Through the use of single-cell genomic techniques, heterogeneity in developmental, timing, and molecular processes that promote proteostasis can be resolved, but the standard practice of pulling embryos makes it harder for these techniques to capture variability in biological replicates. The researchers overcame this problem by combining single-cell combinatorial indexing with DNA oligohashing, which involves short synthetic strands of DNA or RNA to capture transcriptional states and abundance of cell types for hundreds of zebrafish embryos at numerous temperatures. To identify molecular sources and cell types that contribute to the loss of developmental robustness, they studied a large number of replicates in order to infer the source of environmentally induced phenotypes.
Now I'll be talking about methods and figures. In the methods, as you can see, there are many experimental designs used throughout the experiment, such as animal staging and stocking, preparation of bar-coded nuclei from individual embryos, RNA sequence, cell annotation, and so on. I'll be explaining in detail the methods as I explain the figures. Figure 1. Temperature stressed on phenotype. In Figure 1, they show the experimental design of a single-cell RNA study and how they have multiplexed the animals so that they can look at the variation of the individual embryos. They categorize hundreds of individual zebrafish embryos using single-cell RNA to identify the cell types. In Figure 1a, they categorize the embryos in a control group and an experimental group. The control embryos were under normal temperature, 28 Celsius, while the experimental embryos were elevated in temperatures such as 32 Celsius and 34 Celsius. They hypothesized that those embryos in higher temperatures would develop faster. To be able to identify individual embryos, they used hashing with unique barcodes of each embryo. As they conducted the experiment, they noticed the embryos in higher temperatures showed a robust progression of development. As you can also see, there was a push on the genome which resulted in defects such as bent tails. Figure 2. Elevation of temperature causing acceleration of development. The methods being used is cell annotation by projection of data to reference, estimation of transcriptional age. In Figure 1, as we saw, they noticed embryos in elevated temperatures increased in development. Now they wondered to themselves whether all cell types in elevated temperatures develop synchronously. They hypothesized that all cell types develop synchronously and in proper proportion in embryos raised at higher temperatures. Using the reference data set, they analyzed individual embryos in 2 dimensions. Embryos group in similar cell type of composition and trajectory by sample point, as we could see in Figure 2a. They compare the zebrafish at the same developmental stage because it will determine whether they are developing at the same time or not, while being elevated in higher temperatures. We find that some, but not all cell types show altered abundance as a function of temperature. Some cell types show abundance in the notochord, while others show a slower abundance. This suggested that the response to temperature is an integrated non-uniformly across all cell types. Now this raises the question, what is the molecular basis for these cell types to have specific effects? Figure 3. Asynchrony development across cell types. The methods being used here are the estimation of transcriptional age for each cell and cell type in comparison of embryo cell stage with conventional staging. They looked at why some cells were more accelerated compared to others and why some were slower. What's similar about these cell types that are slower? They hypothesized that the difference was based on cellular processes such as cell cycle, protein synthesis, metabolism, or stress response associated with the difference in developmental rate increased among cells. They used cell RNA-seq data, and they found that protein synthesis and protein degradation were more present in the slowdown of cells. Cell types with high expression of genes involved in protein and the unfolded protein response, UPR, showed more temperature-dependent acceleration of development than those with lower expression of these processes.
Figure 4 describes the importance of UPR in the notochord. They used notochord staining and CRISPR to knock out the transcription factor ATF6, which affects a lot of unfolded protein response genes. The hypothesis said that upregulation of UPR and translation related to the notochord sensitivity to temperature. They found out that a significant increasement of enrichment set was detected from those whose temperature was elevated. They also found out that hotspots were found among the epithelial-like sheet cells. These epithelial-like sheet cells produce and process and package a great amount of collagen. Conclusion.
There are four major findings in the study. The first, they discovered that cell type synchrony and developmental rates are influenced by temperature. Additionally, temperature stress transforms the cell types in embryos, leaving long-term imprints that cannot be explained by differences in developmental stages. Moreover, they show that perturbed proteostasis causes notochord cells to be more sensitive to temperature, depending on cell type. By accounting for differential responses to temperature across cell types and integrating individual to individual variation, they identify mechanisms of developmental robustness unique to each cell type. Next, we will talk about limitations and a potential next experiment. Neurodegenerative diseases are caused by an excessive burden on protein homeostasis. The research notes that even transient stress may alter normal development irreversibly during embryogenesis. Temperature or other environmental stressors might lead to further perturbations on homeostasis in cell types with high proteostasis demands during normal development. In future studies of the differential demands placed by each cell type on ancient and general molecular processes, scientists may develop an experimental procedure to gain insight into how stress on a molecular pathway affect tissues, organs, and organismal phenotypes.
Discussion.
At least we want to talk about the ethical issues. So you always have to think about the cost-benefit analysis, which is more difficult in basic research. You also have to think about the 4R principle, which means replacement, reduction, refinement, and responsibility. So you always have to make sure that there's no other way for your research, like a simulation, instead of using animals. But in our case, in our paper, the genome of the zebrafish has been completely decoded since 2013. And the molecular timers of embryonic development are very similar in many creatures, including the human being. Another big advantage was that the zebrafish reproduced repeatedly and reproduced a large number of these in this, and both the egg and the larva of the zebrafish are transparent. So it's easy to observe the individual stages of development and growth. Nevertheless, with the increase in temperature, stress is exerted on the embryo. Are there any questions?